I think we're ready to begin, and thank you all for showing up. What I'm going to do today is do a brief review of the material that I've talked about here at the Women's Conference before, but mostly I'm going to spend time on the papers, the research papers that have been published in the last year. As you are fully aware, this is an arena, scientific arena, in which there is a great deal of excitement. And there are now 2 and 3 papers from very reputable science groups coming out each week. And it's becoming increasingly difficult for me to keep up with it. And I thought that I'd make it easier on you so that you didn't need to take extensive notes. So here's a handout. And thanks to the people that finance this conference, it's in color. I don't normally get to have handouts that are in color. So the theme of today's presentation is going to be weight loss and weight loss medications. But I'm going to pull the same stunt that the weather reporters do on TV. I'm going to tell you that the weather's going to be really interesting, but you'll have to wait till the end of the program to find out how interesting. So we'll start with the really bad history that weight loss medications have had. Back in 1893, when thyroid hormone was just becoming understood, it was the first item that was put on the market to help people lose weight. That's a bad idea, as you can imagine, because what thyroid hormone does, it does speed up the metabolism enormously, but it makes you bug-eyed. And there are several other things that it does that you really don't want to have happen to you. Then there was digoxin. That is the leaf and root of the foxglove plant that is now has a very useful role in treating heart disease, but as a weight loss medication, it wasn't so hot. How many of you took fen-phen when it was popular? Okay, there's a few people here who are indeed survivors of the fen-phen. And you all know why that was pulled from the market? It turned your heart valves into sort of mush. Well, not quite that bad, but the FDA very quickly pulled it from the market after about 3 or 4, well, maybe 6 or 8 months, actually, before they figured out that this epidemic of heart valve problems was due to fen-phen. Dinitrophenol, 1934, that produced cataracts and neuropathy. There have been many other weight loss medications that have been proposed and withdrawn. These were the reputable ones. These were the ones that Big Pharma produced. But there are a lot of weight loss medications in the over-the-counter market and herbal preparations. They've been pulled. One of the most recent ones was ephedra, known as ma huang in Chinese medication. Ephedra comes from a plant that grows out here in the desert. I mean, if you really want some, you can go out there and chew the appropriate plant. But it too had problems. In fact, we know of no weight loss medication that actually has no side effects and does what it's supposed to do. We know of quite a few things that cause you to lose weight, but none that cause you to lose weight safely and without any additional health problems. So it would be very nice if there was such a weight loss medication, and it turns out that there probably is, or there shortly will be, and we've been living with it for much of our lives. But of course, more about that later. So let me talk about the digestive system and the alternative digestive system. You all know about your regular digestive system. It's, yes, wouldn't it be wonderful if we could make our own weight loss medication? Because more than likely, if we made it ourselves inside of our bodies, it wouldn't have any negative side effects. So this is the human body. It's a rather idealized version of it. Those of us that are physicians know that the human body doesn't look quite this sterile, and it's not nice and blue on the outside and pink on the inside. It's not nearly this good looking. But Here we have the entire digestive system with the exception of the stomach, but since it sits in front of some other organs, we pulled it. Soluble fiber and its benefits. Question, how are the main parts of the body categorized? Answer, the body is consisted into three parts: the brainium, the borax, and the abdominal cavity. The brainium contains the brain, the borax contains the heart and the lungs, and the abdominal cavity contains the 5 bowels, A, E, I, O, and U. And that's what we're going to talk about now. This is one of the bowels. This happens to be the small one. And this is the digestive system that you've all been familiar with from childhood. This is where all the food that you eat, the proteins, the carbohydrates, and the fats, get absorbed. Now we're going to come back to this because it's important that you understand that by the time the food gets down here, whether it tasted good or not, whether it looked good or not, it's all kind of a mush. And the only thing that you can absorb is the simplest form of food. So for carbohydrates, the only thing you absorb are simple sugars like glucose. And most of your carbohydrates will turn into simple sugars by the time they get down here. For proteins, what do you absorb? Amino acids, that's right. You absorb the simplest of the building blocks of the food that you eat. And the fats, you absorb free fatty acids, the simplest building blocks. That's the only thing that can go across your intestinal barrier. Now this is going to turn out to be important because before the end of this talk, I'm going to explain why eating the kinds of rolls that you had for lunch today is no different than eating the dessert that you had afterwards. And trust me, the dessert was more fun. So this is the normal digestive system. This is the alternative digestive system that we've only known about for, well, we've known sort of about it for many years, but we've taken it seriously only for about the last 5 years. It consists of the colon and its contents, and the contents are bacteria. This is what you actually look like from the view. If another bacteria were to look at you, this is what they would see. Another bacterium, sorry. Just how many bacteria do we have inside of us? Well, this is a cubic centimeter, and that's my hand, so you can see it's very small. It's 1 centimeter by 1 centimeter by 1 centimeter. And this is 100 bacteria. They don't actually look like that. These are rather cute ones, but imagine for a moment that we've got 100 of them in a square like that, and we're going to make them smaller. And then we're going to put them into this screen, and there. We've got 100 bacteria into 1 cubic centimeter. That's not very many bacteria, even though that's a very small area. So above the ileocecal valve, which I'm going to show you in a moment, the outside, not the inside, we have perhaps 1,000 bacteria per cubic centimeter. There's 1,000 bacteria there in that picture, in a cubic centimeter of the food stuff that's going down your gut. When you cross the ileocecal valve, it goes to not just 10,000. There's 10,000 bacteria. It goes to 1 followed by 12 zeros. Now, I can't guarantee that I've got 1 followed by 12 zeros in bacteria here, but I've got a lot of bacteria there. That's what happens as the food moves through from the small intestine to the large intestine. This is the appendix, in case you didn't recognize it. That's the ileocecal valve. Ileum, this side, cecum, this side. And that's when the food enters your second digestive system. Now, the second digestive system we know relatively little about. And what makes it much more difficult to find out is that it differs in different people at different times in their lives, depending on their diet. So this is sort of a random, it varies enormously, and I'm going to tell you about some of the bacteria that inhabit it that we do know about. But there's at least 1,000 different bacterial species here, and they depend to a very large extent. Which ones we have there will depend to a very large extent on what antibiotics we've been taking recently, what kinds of food we've been eating, whether we've been eating meat or mostly vegetables, or whether we've had a lot of soluble fiber in our diet or very little. So we'll first talk about what we had known up until about 5 years ago. It's important, but I've talked about it before, so I'm not going to spend very much time on it today. Insoluble fiber controls pressure in the bowel. Dennis Burkitt, somebody that I've met on several occasions actually, he's now dead, was a British surgeon in Africa, and he actually did two things that made him famous. He discovered Burkitt's lymphoma, a kind of cancer that is odd because people with malaria get it much more frequently than people who don't. But he also told us something about fiber in the bowel. He noted that Africans in Africa had no diverticulitis, no diverticulosis, or colon carcinoma, while Africans from the same tribe, from the same place in Africa, who had moved to London, had all three. And he was curious. He hypothesized that fiber in the diet was responsible, and he noted that below 10 grams a day, the average in the US is something like between 14 and 17 grams, but there are a lot of people living in the US that get less than 10 grams a day. 85% of the population will have hemorrhoids. 75% will have diverticular disease, diverticulosis or diverticulitis, and colon carcinoma will be common. I'll just show you one of those three diseases. This is the diverticulitis, diverticulosis. What happens here is that the pressure in the bowel gets so high because the diameter of the bowel gets so small and the pressure rises as the contained vessel gets tinier and tinier, that it herniates and you get these little outpouchings through the mucosa. That's the lining of the large intestine. And they pouch out into these little round things, and then they get infected. Here's normal, uninfected diverticuli, and here's infected diverticuli. Diverticulosis and diverticulitis is very common in the US because of our diet, and it's serious. About 30,000 people a year die from diverticulitis. So that's insoluble fiber. That's all we knew until about 5 years ago. And then we realized that in addition to the real fiber in the diet, the sort of stringy stuff that your mother would tell you to eat your vegetables for, that there was something else going on. Now, it got to be known as soluble fiber. And soluble fiber is a misnomer, because in most cases, it isn't fibrous at all. What it is foodstuffs that normally you would think we would absorb before the food got to that ileocecal valve. But because they are molecularly hooked up in such a way that our intestines, our stomach, our liver, they can't be broken down by our regular digestive system, they get all the way to our secondary digestive system. And so mostly it turns out to be carbohydrate that is put together in such a way that our normal enzyme systems can't break it down. There's a fair bit of that in the diet, and there used to be a whole lot more. Back around 1890 to about 1910, when the milling of flour here and in the UK and Western Europe changed from stone mills to steel mills, we lost an awful lot of that because for the first time, millers could take out portions of the grain. With steel mills, I don't know whether you know how milling occurs. I'm sure you think that they take whole grain and grind it up, right? No. They take whole grain, they grind it up, they make 7 different or 8 different, sometimes 9 different streams. And then they combine them at the end. So if you think you're getting whole grain flour or whole wheat flour, you're getting actually a combination of this stream, and of course it's made so that it has a long shelf life, which means almost certainly that a lot of the really good stuff has been left out. The longer the shelf life, generally, the poorer the food stuff is. So French fries that you can put on your table and look at, admire for 3 months, and they don't turn green, they're not worth eating in the first place. Okay, so soluble fiber is not really fibrous. Shouldn't have been called soluble fiber, but the name is stuck. Mostly it's carbohydrate molecules that our regular digestive system cannot break apart. I'm now going to start talking about that, but first I've got to explain what we are doing when we enter the field of dietary substances. I'm sure you have been told that scientists said this and scientists say that, and then they change their mind. And if you just wait long enough, they'll tell you that whatever it is you're doing is really good for you. There's some truth to that, but the reason for that, in foods particularly, and in nutrition, is because of the nature of the data that we work with. Richard Feynman was probably the country's best-known physicist of the 20th century. Scientific knowledge is a body of statements of varying degrees of certainty. Some are most unsure, some nearly sure, but none are absolutely certain. So here we have an emu. It's an explanation most uncertain, or an explanation moderately uncertain, or an explanation minimally uncertain. And you need to know whether you're dealing with an emu 1, 2, or 3, because it makes a tremendous difference how much weight you put on that statement. So, Brian Bull. Scientific knowledge is a collection of explanations of varying degrees of certainty, some mostly unsure, some moderately unsure, a few that are minimally unsure, none that are absolutely certain. And, as you can guess, mostly in diet, we're dealing with emu 3s, explanations that are most unsure. We have a few emu 2s, no emu 1s. Emu 0 would be an explanation absolutely sure, and science doesn't know of any such thing. There's nothing that is absolutely certain. There's a few things that are emus 1. You know, the Earth goes around the sun. We're pretty sure that that's what happens. But, you know, for several thousand years, everybody on Earth knew that the sun went around the Earth because you can see it go round every day. Okay, nutritional science is founded on EMUs 3. Explanations most unsure. It's inherent because every time you change something in your diet, you also change something else. Now, we used to think that was complication enough, but now we know that when it gets down to your second digestive system, what then happens to it depends upon the bacteria that you have down there, and you have very little control over that. And we don't know what most of those bacteria are. Now, the reason that the field has exploded in the last 5 years is because we can't grow these creatures outside of the human gut, but we can do genetic analysis on them, and so we know what their gene, we know what genes they carry, even though we can't grow them. Up until about 5 years ago, we had to grow them in order to determine what we have. Now we don't need to grow them. And that's why we know there's at least 1,000 different species down there. All right, a good microbiome, here's my typical microbiome up there. As I say, they're a lot cuter than the real ones. A good microbiome does things like produce statin-like components. There is a natural statin, it's fairly powerful, that is produced in the gut, and because most of us don't get enough of the stuff that the microbiome needs to produce the statin, we take one. Statins, however, have side effects. Don't have time to go into that today. I talked about those last year. But a good microbiome produces statin-like components, anti-inflammatory agents, anti-cancer agents. I'm going to briefly mention a couple of those. 75% of the nutrition of the gut is actually produced by the gut itself. And it produces an anti-diabetes medication, and of course this one, which I keep promising to come back to later. A bad microbiome produces arteriosclerosis, pro-cancer agents, pro-inflammatory, produces obesity, inflammatory bowel disease, and a whole host of other diseases you don't really want to hear about, but it's how us pathologists earn our living. So, next time you look inside your medicine cabinet, just sort of picture, here's some bacteria putting some stuff in here, and here's Big Farm, and here's your little farm in your gut producing all these wonderful substances, and think of them kindly. These are female bacteria, obviously, with long hair and lipstick, and they're the nice ones. They produce the good stuff that we produce inside of our body. So let's go back now. I'm going to briefly talk about the statins because they have been one of the substances that has been most successful from Big Farm in the last 15 or 20 years. One of the reasons is that the body naturally produces it, or used to produce it in sufficient quantity, doesn't any longer. So the Big Farm has stepped in, and then we now have 13 or 14 different statin compounds. The material that gets through the ileocecal valve and therefore becomes available to the bacteria is metabolized to things called short-chain fatty acids. And the short-chain fatty acids consist of 2 carbon compounds, 3 carbon compounds, and 4 carbon compounds. The 2 carbon compounds are things that you know well. Alcohol is produced in your gut in the amount that's approximately equivalent to a can of beer a day. So whether you drink or not, your liver is quite capable of processing, has to be, because your gut's producing about 1 can of beer's worth of alcohol each day. Now, it's produced slowly over 24 hours, so you're never going to get high on it. And in fact, you don't even know about it because most of it goes straight to the liver and it's metabolized there. But we all have alcohol dehydrogenase in our livers, and that's what it's there for. Well, those are the 2 carbon compounds, alcohol and something called acetate. And it's the acetic acid, the vinegar, the acid portion of that is what's known, you know it well as vinegar. The gut produces a lot of that. I'm going to come back to that. So for the moment, I'm going to go to the 3 carbon one, which is propionic acid. And we've talked about that. Inulin, by the way, is the compound that most of the studies have been done on. It's a polyfructose molecule. It's the same as high fructose corn syrup, except the fructose molecules are hooked together so that it acts like a starch, but we can't break it. And so inulin, almost all of it, gets through our small intestine and into our large intestine, where it's metabolized by the bacteria, and they produce things like propionic acid, alcohol, acetate. It's a short-chain fatty acid produced by bacteria that metabolize the carbohydrate that reaches them. It travels to the liver, and it downregulates a very complex named enzyme called HMG CoA reductase, which is the first step in the synthesis of cholesterol. Now, cholesterol is not the bad actor that you've been told it is. A major portion of every cell membrane in your body is made of cholesterol. So your cells produce it, your liver produces it, you eat a little bit in the diet, it's considered the bogeyman, but the amount of cholesterol you eat in the diet, unless you have a very unusual diet, is small potatoes compared with what your body produces. This HMG CoA reductase is what statins turn off or turn down. And that's why with statins, some people get enormous muscle pain, and occasionally, and this is rare, their muscles just sort of dissolve. I've only seen one case, but that was pretty dramatic. But because statins downregulate this is commercial statins, people who are on statin drugs don't benefit very much from going to the gym. That is, they don't get bigger muscles and stronger tendons because the body can't produce the first step in the synthesis of the cell membranes that are going to be needed in order to do this. So for practical purposes, if you're on a statin at reasonable dosage and you go to the gym, not going to do you any good. It also makes you quite a bit more likely, about the estimates are 30 to 40% more likely to sprain a muscle. And so if we had something that we made ourselves, So we didn't need to take a statin. We'd be better off. And in fact, we do make something. So why don't we make enough? And the answer is very simple. Our diet no longer contains the material that the bacteria used to use to manufacture statins in our gut. But when you give it that, an appropriate amount, it produces this propionic acid in enough quantity that it does the same thing that a commercial statin in reasonable dosage does for you. In case you wanted to see what 3-hydroxy-3-methylglutaril coenzyme A reductase looks like. So much for that. This is just showing you these people with the dotted line. One of them's on statins, and one of them is just getting enough soluble fiber in their gut, and the effect is identical. A bad microbiome produces arteriosclerosis. I'm going to skip over this one because I talked about it last year. Papers came out about 18 months ago in the most reputable available journals, Nature, the New England Journal of Medicine, hit all the newspapers, but to my surprise, an awful lot of people haven't heard about it. We now know why meat is bad for you. We've known that red meat was bad for you for a long time because populations that consumed red meat in reasonable quantities, like 2 or 3 times a week, died sooner than populations that didn't. The difference seems to be about 2 years. But we didn't know why. The facile explanation was that it had cholesterol in it and it was high in saturated fats. But we all knew that that probably wasn't the answer because the amount of saturated fat and cholesterol in meat clearly wouldn't shorten your life by 2 years. So 18 months ago, we discovered something very interesting. Carnitine causes arteriosclerosis, and it's symptomatic of a gut microbiome that's gone wrong. New health worry in red meat. I think this was from the Wall Street Journal. I don't remember. Anyway, major constituent of meat is something called carnitine. Carnitine in a meat eater is transformed in the gut into a really nasty substance called trimethylamine oxide, TMAO. This is Harvard School of Public Health. You've heard of the Nurses' Health Study and the 37,000 male professionals. One additional serving a day of red meat raised the risk of death by 13%. But what we learned then was that if you eat meat regularly, you have 4 different species of bacteria in the gut, and we know what they are. And each one of them does one of the synthetic steps that transforms carnitine, which is a major constituent of meat, into TMAO, trimethylamine oxide, which is a toxin. Now, this explains something that had puzzled us for years, because meat itself could not be toxic. If it was, the animal would have died. But since the animal was healthy until we killed it and turned it into meat, it couldn't have been toxic. So why was it acting like a toxin? And the answer is, this is a bad microbiome in the gut, but it takes this material, carnitine, and synthesizes with 4 steps, and we know what the 4 steps are, and we know what the bacteria are that do it. And they are present only in the gut of people who eat meat regularly. So I gave a lecture a year ago, and my title was, Only a Vegan Should Eat Steak. I thought it was very funny, but in truth, if a vegan eats steak, they don't get this toxin because they don't have the bacteria. So for them, it's perfectly safe. But of course, they wouldn't be vegans then. Okay. So carnitine chemical, not fat, may explain the link between red meat and heart disease, and I'm not going to go into that either. I have already also talked briefly about anti-cancer agents, and we produce in our gut, and now this is by a different synthetic pathway entirely than the ones I've talked about, from a different starting point, but we produce something that looks for all the world like tamoxifen in our gut, provided we give the bacteria that are interested in doing this for us the right food. Now, in this case, it's not soluble fiber. In this case, it is something called lignan, this thing here, or here. It's a fairly simple molecule. It's found in all sorts of seeds. Flax seeds, very rich in it, sesame seeds. So if you grind up flaxseed and add some sesame seed and put it on your cereal every morning, you will get plenty of lignan. The gut microbiome turns it into something called enterolactones, and it is as good as, almost as tamoxifen, and as far as we know, has no side effects. Now, as you're probably aware, women who've had breast cancer and have recovered are often kept on tamoxifen for 5 years, and now they're talking about leaving them on for 10 years. The gut microbiome produces enterolactones, these sorts of things, and you can stay on them for the rest of your life, and as far as we know, no side effects. So, breast cancer and diet is probably an EMU2, meaning the data has come from a wide variety of sources. It's been reproduced in, the last I checked, something like 30 different groups of women. That is, what they do is when the woman is diagnosed with breast cancer, they measure the enterolactone levels and then follow them for 10 or 20 years and look to see how the enterolactone levels correlate with mortality from breast cancer. And it's quite clearly a very protective agent. Dietary lignan intake and breast cancer risk. This is an abstract. This was one of the most interesting ones I found. This is breast cancer mortality over 13 years. I think this was a Danish study. This is survival. And these are the women who had the highest enterolactone levels. And these were above the median, meaning from the middle up. And these were from the middle down. And as you can see, a dramatic difference in survival. Again, the sorts of things you see with tamoxifen. And what's even more interesting is if you followed them for all-cause mortality, the differences were even greater. This is not just measuring mortality now from breast cancer, but from heart disease and lung cancer and everything else you can think of. Just the flax, lignans, and somebody thought they were being very funny when they put that headline together. But this is another study on breast cancer. Okay, we've talked about a good microbiome produces, and I did promise I'd get back to weight control. And that's what we're going to now talk about, appetite suppressant. It's been known that a high fiber diet, and in this case it was thought to be insoluble fiber, the kind of material that Dennis Burkitt was interested in. But it's been known that a high fiber diet is filling, meaning people eat less if there's more fiber in the diet. It was assumed, simplistically as it turns out, that it was just because it was more filling, and if your stomach was full, you'd eat less. Turns out that that's true, but that there are other effects going on in the body, and those have only been known for less than a year. The most interesting paper was published in Nature, and where am I here? Acetate, do you remember I told you that acetates, that's the 2-carbon molecule that's sort of alcohol just kindly sort of rearranged, like it's vinegar. And acetate is known to be one of these molecules that doesn't go straight to the liver. It circulates in the body. So they took rats and they labeled the acetate and then they followed it. And those of you that were in the sleep lecture by Cameron Johnson this morning, he talked about the hippocampus. Well, it goes to the hippocampus, and there it downregulates the appetite. Short-chain fatty acid, it reduces appetite via a central homeostatic mechanism. Unfortunately, when was this? April 29, 2014. So far, it's been done only in rats, but it puts the rats off of their chow really quite remarkably. They start losing weight fairly quickly if you raise the level of this free fatty acid in short-chain fatty acid in the bloodstream. And here's the proof of it. They labeled it. This is before the rat was exposed to labeled acetate. And here it is afterwards. You can see the difference. And it's obviously there's a lot of it in the liver. But what's interesting here is that the location that it settled in the brain is known to be an appetite control center. So that's what we now know. Stay tuned, because it looks very much like we do have perhaps the first no side effects, weight control medication, which you make yourself if you eat right. I'm going to deviate a little bit here to a paper that was published now about 3 years ago, and it's something that I think everybody wants to do, is how to eat more and weigh less. And this is our friends, the emus. The groups that we're going to look at are 3 groups. 2 of the groups were nurses, 1 of the groups were male health professionals, but all of these are well-educated people, followed for anywhere from 13 to as long as 20 years. The Nurses' Health Study, 121,000 registered nurses enrolled in 1976. The Nurses' Health Study 2, 116,000 younger females are enrolled in 1989. And the Health Professionals Follow-Up Study, which was male health professionals, only 51 males to 250,000 females. But we didn't have good dietary data on enough of those to do them all. We only had good dietary data on about 120,000. That's still a very large group. Statistically, we're talking about an EMU 2 here, not an EMU 3 any longer. We're getting serious in this sort of study. And this is what they found. Now, let me explain this graph to you. On average, these people, followed for 30 years, gained 1.5 pounds a year, on average. And that 1.5 pounds a year either went up, or they stayed the same weight, or they went down. And what they looked at, because they had good dietary histories on these 110,000 people, what they looked at was the change in diet and whether your weight went up or down as a result. So these are the foods, not too surprisingly, if you ate an additional serving a day of potato chips, then on average you would pick up 2 pounds a year. Fair enough? Nobody finds that surprising, do you? Well, equally bad was fries. Potatoes or fries. Now, this is not boiled potatoes or, properly cooked potatoes. These are, potato-flavored fat chips, because most of the calories in a potato chip is coming from the fat. It's not coming from the potato. So this is not saying potatoes are bad. It's just saying that fried potatoes with salt are bad. Now, canned meats, not too surprising. Unprocessed red meats, not too surprising. Butter is surprisingly little. An extra serving a day of butter only gave you like 1/4 pound a year on average. But what is really interesting is that certain foods caused you to lose weight. Vegetables. Now, why would eating vegetables cause you to lose weight? Well, obviously, you only have a certain amount of, capacity. And if some of that's filled with vegetables, you're going to have cut something else out. So every additional serving a day of vegetables caused you to lose not a great deal of weight. That's maybe 1/2 pound a year. Nuts, maybe a little better. These different colored bars are these 3 different populations, the males and the 2 female populations of nurses. One was younger than the other. They were all nurses. One of the reasons they stuck with those is because these people do a much better job than people who are not well trained to follow their diet. I mean, they know what you get much better data from them.
So was it the younger or older women who ate more nuts?
Well, all 3 of them are shown here. Oh dear, I can't tell you. Anyway, no, I don't know. Now, I want to show you some very interesting stuff, though, because these whole grains, fruit, yogurt's way on this side. And then back down here, we've got beverages, sugar-sweetened beverages. That's not at all surprising. 100% fruit juice, not too surprising because there's a lot of sugar in it. Whole milk actually caused you to lose weight, and skim milk caused people to gain weight. But the data here is so close to 0 that for practical purposes. So all those years you've been drinking skim milk, it didn't hurt you, but you could have been enjoying whole milk that whole time, and you wouldn't have gained any more weight. Now, this is an illustration of the difference between the known digestive system and the one we've just discovered, because a lot of the things that are going on here are going on because of this newly discovered digestive system. I'm going to come back to here. So, okay, here we are. The NHS, these are the women that have been followed for about 30 years. So that's that bar. These are the younger ones, and then these are the men. Okay, let's look at this. You notice that refined grains are on this side. You notice that whole grains are on this side. Now that in itself is fascinating because what it's saying is that it's not just the number of calories that you're ingesting. It's the other stuff that's coming with it that's affecting your second digestive system that is doing some dramatic differences. So for every slice of bread that you eat that's made from the white bread, well, consider the rolls we had for lunch today. That's a classic case of bread made from refined grains. That puts weight on. Okay, maybe that's about a pound a year, but over 30 years, that's 30 pounds. Whereas if you'd had the same thing in whole grains, you would actually have lost weight. So calories count, but sometimes they subtract. They don't add. Let's look at something else here. This is fascinating stuff here. Sweets and desserts and refined grains are treated by the body identically, except that sweets are much more fun to eat, right? So if you feel good about yourself for eating bread and rolls instead of desserts, you've been fooling yourself. You could have enjoyed the desserts and it wouldn't have made any difference. That's not entirely true because sweets do tend to do a number on your teeth. But if you ignore that, by the time we know that this has got to be true because by the time that these refined grains get down into the intestine, they're all simple sugars, just as the sweets and desserts are simple sugars. The body can't tell the difference. It has to be reduced to glucose, fructose, and other simple sugars in order to be absorbed. So if you feel good about yourself by turning down the dessert and eating an extra roll like we had at lunch today, as far as your gut's concerned, it doesn't make any difference. It's all going to be glucose when it goes across into your body. Oh, other interesting things here. Sugar-sweetened beverages. Oh, yogurt. Yogurt's a fascinating story, and I don't have time today to go into it. That would be pretty much a one-hour lecture all by itself. It's known that the critical component of yogurt is the bacteria. But it's not as simple as saying that if you eat a lot of yogurt, you're going to change your microbiome to a more healthy. You probably will. But in rats at least, yogurt's effective even if you kill the bacteria in the yogurt before you give it to the rats. And it looks very much like something peculiar is happening in the immune system of the gut. Because if you take the lymph nodes out of the rats from the intestine and look at them, they have a lot more immune cells in them if the rats have been fed on the bacteria that are found in yogurt. So the way they do this is they take the bacteria out and put it in the rat's drinking water. And they work just as well as the yogurt itself. But yogurt is one of those delightful foods that's easy to eat, and people enjoy it. And the more you eat it, the more likely you are to lose a couple of pounds a year instead of gaining a couple of pounds a year. As far as we know, it doesn't matter whether it's high-fat, low-fat, or no-fat yogurt, and trust me, high-fat yogurt is much, much better. But it's very difficult to find nowadays. Essentially, all of the yogurt that you get is going to be low-fat or no-fat. But I'm going to talk now about diet. Now, you all, how many of you here drink diet beverages? Oh, come on, I'm sure half of you do. No? 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 Oh, okay. Well, I've got a very select group here. Diet sodas, of course, have non-caloric sweeteners in them, and people drink them because there's no calories, but they taste good, okay? It's not okay. Saccharin and all of, at least the ones they've checked, which is aspartame and sucralose, I think. All three non-caloric sweeteners do a number on your gut microbiome. And it's bad in two different ways. First of all, it changes the microbiome. And if you put rats on saccharin, I'll show you some. The data was on, most of the data was on saccharin, but they checked the others, and to the extent that it was possible to do the studies, they look like they all behave the same way. They change these bacteria, these drop by two log orders, and these go up. Clostridioides, you know what the C stands for in C. difficile? It's Clostridium. This is generally speaking not the good bacteria. So they took and put rats on non-caloric sweeteners, and saccharin, sucralose, and aspartame. And I think this is 12 weeks into the study. They are all got a diabetic or now a pre-diabetic glucose tolerance curve. And all that they have done is just put them on the maximum daily dose allowed by the FDA. Okay? This is not huge quantities. This is what a person would get if they drank a lot of non-caloric sweeteners in their beverages. The normal glucose tolerance curve, when you take and inject glucose, they inject it in the rats. For humans, we let them drink it. It's nicer. This is a fairly normal glucose tolerance curve. Starts here around 100, may go up to 200 briefly, and then drops immediately back again. But this large curve here says these rats are pre-diabetic, and we made them this way in 12 weeks just by giving them non-caloric artificial sweeteners in the dosage that the FDA allows in human beings. Now, what the FDA did when they allowed people to put them in the market was they checked them for toxicity, and these things have no toxicity. But they change our gut flora, and then to prove that this was the case, took germ-free rats, transplanted the gut microbiome, all these bacteria now that had developed on the rats that were on the artificial sweeteners, and produced exactly the same glucose tolerance curves in them. So we know that it's transplantable into germ-free animals. Well, we're pretty sure it's the change in the microbiome that you can't do much else. That's about as, so this is why this is getting to be an EMU2. Saccharin and glucose. Glucose, of course, is, they have to have a control. So they either put glucose in the drinking water or they put saccharin or aspartame. And here's the studies with the donors. So that blue line says that these were germ-free animals. They changed the gut microbiome by transplanting it into germ-free animals and then followed them. And now you think that's bad news. This is even worse. What happened here is that when they looked at those organisms, glucose, this is the control group, and this is commercial saccharin, they discovered that what they were doing was changing the microbiome in such a way that it was able to wring more calories out of the same amount of food. So, okay, the artificial sweeteners do not in themselves turn into calories, but they change your microbiome so you become pre-diabetic, and you now start wringing more calories out of your food. So for practical purposes, you're worse off in two ways. So if you can squeeze more calories out of your food, you're going to get fat on the same amount of food. Any questions arising out of the handout? If not, thank you.